You.
All right. All right.
All
So,
right, thanks, guys.
all right. Welcome, everyone. Thank you so much for being here. It's truly an honor. Thanks for coming to my virtual defense. So let's just get right into it. So when I'm talking about this research project, I always like to start with this image. This was actually an image I took in my friend's airplane. I'm very fortunate that I have that resource, that I know someone who's a pilot. So this is just a great image showing the Ocean City and Assateague Island coastline. So this is near and dear to me because this is where I spent most of my days as a young child. I grew up here. And what's so interesting about this photo is it shows a unique perspective of how dramatically humans can alter natural landscapes. So at one point of history, this was just wherever in the 1930s, there was a huge storm that wiped out this section of this barrier island. And this is the inlet that flows today. And before that time, they had plans, they had it all mapped out. They were going to commercialize this area that's federally protected now. So as you can imagine, if there was this human intervention, it would look just like Ocean City. So what my studies are questioning and are focusing on are exactly how does human intervention and landscape management affect these types of areas. So there's a lot of studies that look into the macroecology and are understanding how dramatically they can change these environments. So whether it's bird populations, amphibians, reptiles, things of that nature. However, different environments are well understudied, and the environment that I'm particularly interested in are the soil environments. So what's going on below ground? And specifically the organisms that I'm interested in are the microbiota. So all of the fungi, bacteria, and archaea that are growing in these soils, how are they being impacted? And how is this gonna affect above ground ecosystems as well? So why would we be concerned with soils? Why should we even care? So soils really provide the basis of ecosystem function. Nothing can happen above ground without that base. So soils are going to be providing a living matrix for microbes, plants, and animals. They're going to act as an important carbon sink for to mitigate climate change. So as Dr. Brian was mentioning, I did my undergrad studies in his laboratory, and we were really focusing on physical and chemical properties of soils within the Salisbury University Arboretum. So we actually were able to quantify the total organic carbon that our university was sequestering. And it turns out on just our relatively small campus alone, we were sequestering at the time 369 tons of total organic carbon. So 1 ton is around 1,000 kilograms. So we're really, it's really acting as an active carbon sink. Also, they're going to be working to absorb and filter excess stormwater runoff, and they can act as a sink for anthropogenic toxins like heavy metals. However, the functionality of soils are inextricably correlated to the microbiota that inhabit these areas. So I'm talking about microbiota such as fungi, bacteria, and archaea. So, and these are the populations that I'm really interested in this study. So how may different land management practices be affecting these communities? So just a couple differences between these microorganisms. So bacteria and archaea are mostly single-celled organisms, and they're all prokaryotic. Fungi are mostly multicellular organisms, and they're eukaryotic. And I also wanted to point out with fungi, everybody, when they think of fungi, they think of mushrooms, right? Well, that's only a short span of their life cycle. They spend most of their time growing throughout the soils in this netted venation-like structure. This is called mycelium, and these are going to be found throughout soils globally. So I really like to refer to them as the internet of the soils. So why would we be concerned with these soil microbes? So I like to refer to these environments as your soil microbiome. So many people are familiar with the human microbiome, right? So we have microbes that live within our bodies that we are dependent on. where we live in symbiosis with them. And it's going to be the same thing with these soils in different environments. So, and what are these microbes going to be doing in these soils? So they're going to act as decomposers. So they're actually creating these soils. This is a great image from Paul Stamets' book, Mycelium Running. And it's showing this fungal mycelium working to break down just these wood chips into soils and really making that environment for other organisms. Also, they're going to be imperative for symbiotic relationships. So it turns out that 90% of angiosperms, flowering plants, form ectomycorrhizal symbiotic relationships at their roots. So basically, these fungi are providing nutrients for these plants in return for a carbon source that's photosynthetically produced. Also, these microbes are going to be driving biogeochemical cycling, so the nitrogen cycle, the carbon cycle, and a particular benefit for humans is they're going to be providing many medically and industrially relevant products. So just to give you a little bit of a perspective of how intense these microbes are going to be affecting these soils, 1 gram of soil contains around 1 billion microbial cells. So that's going to amount in mycelium around 1 mile of mycelium. So just an expansive amount of growth. So just this hiking boot alone is probably covering around 8 miles of this mycelium venation. It's just absolutely incredible. So what's the soil environment we're studying? So we are studying soils in the Salisbury University Arboretum, and I just want to give you a little bit of a historical content, a blast from the past. So this is actually a picture from 1928 of mules excavating the basement for Holloway Hall, literally mules. So this is way before any sorts of huge industrial machinery was being used. Then we fast forward a little bit. This is in the 1950s. So this was the first standing building on campus. This is Holloway Hall. And as you can see, just to give you a reference, this is West College Avenue. This is Camden Avenue, so on and so forth. And as you can see back then, there weren't really too many buildings or cars or different structures on our campus. Most of it was pretty much barren, void of trees, and it was purposed for agriculture. So there was a lot of strawberry and peach farms within these areas. So we fast forward a little bit, and this is a picture from the 1970s. And as you can see, there's more buildings and parking lots and cars and things of that nature. But also during this time span, this is really when we started to build up our arboretum. So what an arboretum is, it's going to be a place that's devoted for a tree museum. So they're going to have a certain diversity of trees and a certain amount of trees. We have over 150 tree species on campus with over 3,000 trees now, which kind of looks something like this. So this is an incredible photo right here. I actually took this in my friend's plane as well. We took a flight right over the campus, and I love this image because it literally parallels this photo from the 1950s, and you can really see these differences. So as you can imagine, as time goes on and population densities increase, this human intervention is going to be exacerbated as well. However, the silver lining behind our campus and the creation of the Arboretum is we've done a great job of preserving these more so natural landscapes. So in addition to elucidating if our campus is harboring microbial diversity, an immense amount of microbial diversity, we want to see if different landscapes are having an effect on this microbial diversity in these soils as well. So what are our hypotheses? What were they going into this study? So we hypothesized that microbial diversity and composition would differ among managed landscapes across the Arboretum. We were looking to determine that physicochemical properties such as pH will be strong determinants of microbial assemblage across Arboretum soils. And specific microbial taxa may be used as indicators of soil condition. And I just want to emphasize on this point here, because usually we use physical and chemical properties to assess a soil's condition. However, in the advancements of technology, we're starting to see evidence that we may be able to use certain microbes to do the same job. And this may be even more responsive and effective, because if you think of a biological system, they may be able to respond to certain anthropogenic perturbations in a more effective time manner for assessment and management. So when I talk about I just want to talk about how we classify these different landscapes across the Arboretum. So we categorize them into 4 basic landscapes, and it's going to be based on the intensity of human intervention, the relative intensity of human intervention, and also basically the above ground plant flora, some of those characteristics. So I'll just kind of go through these. So one of the first landscapes we categorized were lawns. So this is an example of a lawn. And as you can see, it's going to be consistent of monocultured grasses. It's going to be pretty much completely void of trees. There's going to be a lot more foot traffic in these areas, etc. And then we have these active gardens, and these are going to be really diverse across campus in the Arboretum. So they're going to be consistent of small trees and shrubs and different ornamentals, and they're going to be heavily intervened by human management. On the other hand, we have sections on campus that are relatively undisturbed. So we describe these as forests, but I just want to note, we make the discernment that they're undisturbed pine forests because as you can recognize in the Salisbury area, loblolly pine is really the dominant tree species in our area. And also we have these parks that we describe as well, and these are going to be managed tree and turf grasses. And a lot of our campus are consistent of these parks because the campus is nested within the Arboretum. So along with building the campus, we built this diversity of trees. So these parks are really going to be in between in terms of human intervention and plant flora between these forests and these lawns. So as you can see, there's going to be some greater growth trees in these areas. However, they're going to be tending to these monocultured grasses. They're going to have little mulched beds around the bases, so on and so forth. So this is also to the right here, this is a map provided by the Geography and Geosciences Department. And this is really just showing the allocation of these different areas. So this lime green areas are going to be these parks. Forests are going to be these dark green areas. These brick colors are going to be representing these garden beds, and these lawns are going to be represented by this somewhat tannish yellow color. And as you can see, I'll just repeat myself, the campus is nested within the Arboretum, so you can see that these park areas dominate the landscape that supports Salisbury University. And I just want to make one note because this map needs to be updated. However, this was one of our sampling sites in front of the bookstore. And right here to the left, there's a little path that separates us, but this is another forested area. However, this is really not an undisturbed forested area anymore. This is, they kind of made some pathways in this and they actively manage it. It's a butterfly garden now. So we actually, we didn't want to lose one of those forested sites. So we actually sampled at a location across from campus. So right across Camden right here. And this is Dr. Folkoff's property. He used to be a professor in the geology department. So I just wanted to make that clear. So if you're familiar with the Arboretum, you might recognize some of these places. And I just wanted to give you a little bit of a visual interpretation of these different landscapes that we sampled from and the microbiota that we're interested in these landscapes. So this is the pergola gardens. We have the Folkoff forest that I was just describing. This is Fulton Park, GUC Park, Henson Park, and everybody should be familiar with this iconic image of Holloway Lawn. So we're interested in soils, right? And the microorganisms that inhabit them. So we have to collect soil, right? So I just want to describe our sampling methods a bit. So we took all our soil samples on the same day in April just to minimize weather variation. And how we did this was, I like to call this the lightsaber of the soil scientist. So we use a 2-centimeter steel sampling core, and you basically just get a soil profile like this. However, we're only interested in the top 10 inches of the soil, so more so this organic matter, because this is really where the microbes are going to be most active and reside. And between each sample, what we did a 70% ethanol rinse to hamper biological cross-contamination. We are dealing with microorganisms, so we don't want to cross-contaminate these samples. And we took samples from 14 different sites on campus. And at each site, what we did to maximize the representation of the whole landscape is we blocked each landscape into 4 equal sections, and then we took 5 soil cores from each section. 5 soil cores from each section. And each of these soil cores, what we did was we homogenized them and we pulled them together. And we did this to cut down on sampling costs while still representing the landscape as best as we could. So each of these 14 sampling sites had a total of 4 pooled samples per site. So if you just look at the work that we put into it, this was a total of 280 cores, and after pulling them together, it was a total of 56 samples. So it was a huge sampling effort, and I thank all the undergrads that helped me with that, and professors as well. This is actually a picture of Dr. Elizabeth Emmert at work. So what we did was we used nitrile gloves for sterility, and we transfer the soils into these sterile Whirlpack bags. So what did we do with these soils after we collected them and put them in the lab? So in the field, we put them on ice, and then we put them in a freezer at minus 20 degrees Celsius before we did anything with them. So how are we going to study these microbial communities in these soils? So what we used was a DNA barcoding method, and this is synonymous to barcoding something at the grocery store. So say I have this chapstick and it has a particular barcode, you scan it, it goes to a database, and then it brings you back information about that particular item. And that's exactly what we're doing with these microbes, but instead of using this actual barcode, these lines of black and white, we're using its DNA to identify these microbes. So what do you have to do first? First, we extract DNA, so we just use a basic kit to do that. And then we use polymerase chain reaction to isolate a certain genetic markers. So as I said earlier in my talk, there's around 1 billion microbial cells in a teaspoon of soil. So this is going to be a lot of DNA and a lot of information. And we don't necessarily need all that DNA. We just need a little tiny barcode. So polymerase chain reaction, it's synonymous to picking up a book. And if you just want 1 page of that book, you rip up that page, you put it on a copier machine, you make millions of copies. This is what we're doing at the molecular level with this DNA and using these certain biogenetic markers. So after we isolate those markers using PCR, we used gel electrophoresis to make sure that we're getting the actual size that we want, the actual product that we want, and that we're actually getting a PCR product. So this is an image of one of the gels we ran, and it's going to be excluded based on size. So what we were looking on, just for example, with bacteria and archaea, was something around 250 base pairs. So then we use what's called nanodrop to quantify and qualify this DNA before we send it off to be read, because we want to make sure we have a PCR product of good quality. And so we sent these off on dry ice to Michigan State University Genomics Core for next generation sequencing. So they're going to be able to take these genetic markers, they're going to tell us exactly what the sequence of those are, and then send them back the information for us to process. Also, what we did was we did physicochemical analysis as well. So things like pH, micronutrients, macronutrients, bulk density, things like that. But we were able to package and send those out to agro labs in Milton, Delaware. So when I was an undergrad, I was really doing these sorts of analyses in the lab, but thankfully I was able to gather up enough money, and I was able to just focus on preparing the DNA and focusing on deciphering these sequences. So I just want to go into a little more detail about the genetic barcode that we used. So for bacteria and archaea, so these prokaryotic organisms, we use what's called 16S-V4. And 16S is just referring to a ribosomal subunit. So the gene that we're getting, so we're amplifying RNA from a particular gene within this DNA, and this is going to be a ribosomal gene. And why you want to use, why a ribosomal gene will be good, ribosomes are used to synthesize proteins, right? So every organism is going to be synthesizing proteins. Therefore, we can expect this gene to be in every one of these prokaryotes that we find in the soils. So there's different variable regions that you can use on this gene. We use variable region 4, and so this is just a little picture of that. On the other hand, we actually use a different biomarker for fungi. We use what's called the internal transcribed spacer. And this is a region of DNA that's between two subunits. So this is 18S subunit, this is 5.8S subunit. We use ITS 1F and ITS 2. So these are going to be slightly different genetic markers, but they're going to be amplified from a ribosomal gene. And I just want to point out and emphasize as well, most of these studies, these microbial surveys, they usually either look at just the prokaryotic organisms in the soil or the eukaryotic organisms in the soil. So this really adds power to our study that we're doing both of these. It really makes it unique. Just to add as well, so in addition to sampling and identifying what's going on in these soils, we made these mock communities. So we want to make sure that our next generation sequencing methods are precise, right? So what we did was we made little mock communities, little concoctions of microbes that we know are in the sample, and we sent those off, and we're really testing to see if we can accurately identify something that we know that's in there. And we actually were able to identify all these organisms, whether they were prokaryotic or eukaryotic. These are just some petri dishes of me growing up the eukaryotes in the lab. And I want to give Dr. Patty Erickson the credit for the prokaryotic end of this, because she did the grunt work and she made this mock and she just gave me the mixture of DNA. So thank you for that. So what do we do with all these sequences when we receive them? They're just going to be in a file, and they're just going to be A, T, C's, and G's, right? Just the basic acronym of DNA. However, to process these, we use bioinformatics. So we use a program called R, and specifically there's different pipelines. You may have heard of different ones like KIME or Mother, things like that, to process these reads. We used a relatively new pipeline called Data 2. And so for bacteria and archaea, these are prokaryotes. Just like I said with barcoding something at the grocery store, you're sending information to a database. We're going to be sending information to these ribosomal databases. So for bacteria and archaea, we use what's called SILVA. And this is the high quality ribosomal RNA databases. And the initial reads that we got back from sequencing was 8.5 million reads. And then after processing them, we were left with 5.4 million reads. And we were able to identify through this, by comparing it to this database, 57,000 ribosomal sequence variants. So that's going to be an individual amplification, right? That's what we're talking about with the ribosomal sequence variants. So this is ideally its own microbial taxa. On the other hand, with fungi haven't been described as long. as what these bacteria and archaea have been. So just take that into an account with the robustness of the databases. So we actually had 10 million initial raw reads. We were left with 7 million after processing, and we were still able to infer 23,000 ribosomal sequence variants. So just a breadth of information. And I just did this for a fun little calculation. So each of these ribosomal sequence variants, the amplification, the genetic marker that we isolated, on the low end are around 250 base pairs. So I calculated that we were going through 4.68 billion nucleotides of information. So in the Guinness Book of World Records, the largest book is only around 9 million characters. So it trumps that. And so it's just amazing how much information we can use and process by utilizing the power of bioinformatics. So just to go over an overall schematic of what we did. So we took soil samples, we extracted its DNA, we isolated these genetic markers, we sent them off for sequencing, we used bioinformatics to decipher these sequences, and we come up with a microbial assessment. So what are we specifically trying to understand about these microbial communities, right? We're trying to identify them, but we want to see if the diversity is different and what may be driving the differences in microbial assemblage and diversity of these microorganisms. So just to work with a couple of definitions before I get into the real data analysis of this project, I just want to pinpoint some terms. So when we're studying these microbial communities, we're going to be studying richness, alpha diversity, and beta diversity. So what richness is how many microbial taxa are found in a specific sample. So if we just look at richness, so this is community one, just pretend this is a soil community. This is just a great image to use. It just happens to be a forest. So in this community, there's 4 different taxa. So taxa A, B, C, and D. So the richness would be 4. So if we look at community 2, so this is just say this is a different soil sample, we see that taxa A, B, C, and D are all there. So if we just look at the richness, we might say that these communities are actually very similar. However, diversity takes into an account the evenness of each one of these species, so or these particular taxas. So taxa A is composed of 25% along with B, C, and D. However, in this community, taxa A is dominating. It's 80% of this population. So if you're looking at the diversity of it, these communities are actually not quite similar at all. So alpha diversity is just looking at the diversity within one community, and beta diversity is looking and comparing the differences of this diversity between multiple communities. So first we want to know how many different microbes are in the soils, right, across these different landscapes. What's the abundance of these microbes present? And is the microbial assemblage different between these communities found in park soils, lawn soils, garden soils, or these forested areas within our campus? So just for any of you who aren't too familiar with taxonomy, this is the basic hierarchy of how we identify organisms. So the greatest is going to be domain, kingdom, phylum, class, order, family, genus, species, and it's going to become more specific as it approaches the species level. However, even though there's been advancements in this technology, in most cases, we're not going to be able to identify things all the way to the species level. We're lucky if we can identify them even to the family or the genus in most cases. So just take that into an account when I'm talking about specific microbial taxa. It can be either the phylum, class, order. It's not talking about an individual species. And that just goes to show that we really need to keep at it with these microbial surveys so we can identify more taxa all the way down to the species level. So what do we find with richness? So how we marked these differences between these communities, we used a statistical test called a one-way ANOVA, and these are going to mark the differences. However, it's just going to show that there's a basic difference between these different classes, but it's not telling what landscapes are significantly different from one another. So to do that, we use Tukey's post hoc analysis, and this is represented by having individual letters. So just to go over this, so in terms of richness, when we're talking about prokaryotes, it looks like garden landscapes are going to be the most microbially rich. So they're going to have the most amount of taxa in those areas, followed by lawns. However, lawns aren't significantly different from gardens or parks because they share a letter. And forests are going to have be lowest in terms of richness. And just to put this out there throughout the talk, our p-value, our significance level is going to be anything under 0.05. So for eukaryotic organisms, these fungi, the only trend that we see are that these forests are actually lower in terms of richness than any other landscape across campus. However, just to remind you, we are just talking about how many taxa are in our particular landscape at this point. So we want to incorporate the evenness of all of these different microbial taxa to get a sense if the diversity is different as well. So bacteria, how we calculated diversity, we used the Shannon-Wiener index, and basically the higher the Shannon-Wiener index is, the more diverse that community or that landscape is going to be. So we see the same sort of trends that these gardens or lawns are actually more diverse in terms of bacteria than these parks and these forests. Forests are going to have the lowest diversity on our campus. Then if we separate just the archaea, we're going to see kind of the same sort of trend. Actually, lawns are going to be the most diverse in terms when we're talking about archaea. Forests are still the lowest, followed by parks. And for fungi, we see a trend that these forests and parks are actually lower in terms of diversity than these garden and lawn areas. So this is just taking into account the diversity of these different landscapes. However, we want to know if the actual microbial assemblage is different amongst these different landscapes. So how did we elucidate that? We use what's called a permutational multivariate analysis of variance, for short, a PERMANOVA. And this is going to be a non-parametric equivalent to a MANOVA. So this is going to tell us we're going to be able to use class, so these different landscapes, and see if these microbial communities, the actual assemblage of them, what's making them up, what taxa is different across these landscapes. So to visualize this, we use what's called non-metric multidimensional scaling, so NMDS. And basically, this just takes all the information of the differences of these communities and collapses them into 2 dimensions. So it's something that we can visualize and actually comprehend. We can't really comprehend anything past the 3rd dimension. So these are all color coded. So forests are going to be red, gardens are going to be green, lawns are going to be blue, and parks are going to be purple. And how you interpret this plot is samples that are more tightly clustered are going to be more similar, as opposed to samples that are more spread apart, they're going to be less similar. So these are individual samples. And as you can see, what we expected that these different microbial assemblages are going to be different throughout these landscapes, you see this sort of tight clustering. So you see the gardens clustering in this section here. On the other end, you see these lawn communities, followed by these parks and these gardens. They're all clustering individually. And also these rings represent 95% confidence intervals. So we're 95% confident that a particular microbial assemblage is going to fall within these certain sectors on this NMDS plot. And this is exactly what we wanted to see. So just to give you some perspective, these forest areas are here, lawns are all the way on the other end, and these parks that we kind of described as an intermediate in terms of plant flora and human intervention, they're going to be kind of smack dab in the middle here. So this is for the beta diversity for bacteria. So for fungi, we see pretty much the same exact thing. However, these communities are actually separating in a more intense degree. You can see that these 95% confidence intervals barely overlap. So this shows good evidence that in addition to the diversities being different, the actual microbial assemblage is different amongst these landscapes in these fungal communities. Now, on the other hand, with Archaea, what did we find with them? So with the Permanova, we did find it significant, but however, just like in ANOVA, this is only going to show that there's some sort of difference. It's not going to emphasize what classes are significantly different from one another. So that's why we use non-metric multidimensional scaling. So as you can see, this is not what you want to see if you're trying to prove that these microbial assemblages are significantly different. All of this is kind of, you know, like a shotgun blast, and it's all over the place. However, maybe what's driving the difference in this Permanova is that these forests look like, you know, there's not clustering anywhere near these other communities. However, it's not too convincing because they're not clustering tightly together. So just for terms of being concise for the rest of the talk, I'm really just going to be describing the bacteria and the fungi for the rest of the time, because any other analysis beyond this really wasn't significant with these archaeal communities. So now we've shown that species richness varies amongst these landscapes across campus. Also, the diversity is going to be different amongst these landscapes. And more descriptively, that these microbial assemblages themselves are different. So what is actually driving the differences in these microbial assemblages? What taxa are going to be responsible for this? And what environmental perturbations may be driving the differences in these communities as well? So like I said, we did physical and chemical analysis to describe these different soil communities as well. So to understand what variables may be driving the differences in these communities, we fit these variables to these microbial assemblages. So we use what's called canonical correspondence analysis. And basically, it just maps any significant environmental variable that's actually correlated with the structure of these communities. So as you can see, these forest soils are clustering here, so they're going to be consistent of a lower pH. Maybe higher aluminum concentrations are going to be more descriptive of these park communities. Higher potassium levels are more descriptive of these lawn communities, and maybe higher levels of magnesium and organic matter are going to be responsible for the assemblage of these garden communities. So this is for bacteria. This shows great evidence that different environmental perturbations may be driving the assemblage of these microbial communities. And also we did the same thing with fungi. It's not as convincing, but you still see some of these same sorts of trends. So higher pH is going to be responsible for these garden and lawn communities. We see again with these fungal taxa, these lawns may be driven by higher levels of potassium, and inversely related, these parks may be dominated by microbes that are tolerant of lower pH. So now that we're starting to produce this story that these different environmental perturbations amongst these landscapes are driving these microbial assemblages, what microbes are being affected, and just how we may be able to use different physical and chemical properties to describe these communities, can we use different specific taxa to describe these environmental perturbations as well? So just to give an overlook before I go into the taxonomy of what we found in our soils, these are just going to be the dominant phylum of bacteria, archaea, and fungi. So you can see these are stacked bar plots. This is relative abundance, 0 to 1, which would be 100%. So we have dominating our soils in terms of bacteria or protobacteria, acidobacteria, actinobacteria. In archaea, these Thermarchaeota are dominant, which is commonly found in these microbial surveys. And in fungi, they're going to be dominated by Ascomycota and Basidiomycota and other microbes alike. So we want to determine if specific microbial taxa can be used as indicators of different environmental variables, right? So what we did, we used what's called a Lefse's analysis, so linear discriminant analysis of effect size. And basically, this is going to emphasize if the relative abundance of a particular microbe is enriched based on a specific class. So the class that I'm talking about, of course, is the classes that I've been talking throughout the whole seminar. So different landscapes throughout campus, forests, gardens, lawns, and parks. So if we just look at this, we can maybe use the different microbial taxa to infer these environmental variables. So this is a novel taxa, the order Galeales. Maybe these are going to be descriptive. Maybe they're going to only be able to reside in soils that have higher aluminum concentrations. The same thing if you go through other landscapes. So Delta Proteobacteria, this is a specific class of bacteria. Maybe they're more so described by environments that have higher potassium concentrations. And if we go down, this is, so these are all, so CCD24, PLTA, these are all novel taxa. So just to take into account, only around 1% of microbes are actually culturable. We can grow them in the lab. So this is what makes these types of studies so important and so useful, is that we don't need to grow them up in the lab to understand what's making up the soils, but we're not going to really know much about them at this point. That's why these studies are really important to continue and to build upon. And then also, I love this. You can just see, so just for example, the phylum Acidobacteria is enriched in these forest communities. They love acidic environments. So, and you can see that these different microbial assemblages in forest communities are described by lower levels of pH. Also, we see the same sort of thing for fungi. So different taxa may be able to be used to describe these different communities across campus. So, and in some cases, we were even able to go all the way down to the genus level of enrichment in these particular environments, which is more so important to become more specific about this analysis. So then we want to think about what environmental actions may be affecting these different soil properties, right? So pH is affected, these higher pHs. So these are more so towards neutral pH because we're actively managing them, right? And however, in these forests that are dominated by pines, as they drop their needle foliage, it's been proven that this foliage from these loblolly pine needles actually makes the soil more acidic. So, and in different areas of campus that are being managed, so in these garden communities, when they're continually adding organic matter and fertilizers and things like that, you're going to see differences in the physical and chemical properties that are going to drive the differences in these microbial assemblages, right? Just like we look for particular characteristics on where we live and where we can inhabit, so do these microbes as well. And they're really sensitive to this environmental impact. So what are the overall conclusions of our study? So the Salisbury University Arboretum our soils harbor a high degree of microbial diversity. Both bacterial and fungal communities significantly differ amongst these landscapes in terms of richness, diversity, and microbial assemblage. And we actually found on our campus that particular bacterial and fungal taxa are enriched in these different landscapes. So they may be able to be used as indicators of soil condition, which is important because they might be more effective and more precise in the long run than, say, actually studying these physical and chemical properties that take a long time to actually show these different levels of concern. So some future ideas that I have for this project, if it's continued, that can enhance the power of the study, is making qPCR probes for particular taxa. So right now, these probes that are made for quantitative PCR are only specific enough to amplify these different ribosomal genetic markers, so just 16S or ITS. But I think it'll be useful if we can come up with probes that actually can amplify a specific taxa, so we can actually quantitate and not relatively quantitate a particular taxa to a certain environmental variable. And also, we want to study the genetic ontology to elucidate functionality. So just because a particular microbe is in the soil, it doesn't really tell us the complete story of what it's doing. So we want to use different levels of genomics, so metabolomics, proteomics, to actually elucidate how these are functioning in these soils and maybe impacting the overall function of the soil itself. So with that, I just want to do a few shout outs. I want to thank my committee. Thank you for all your support. This has been quite a journey, and I couldn't have done it without all of you. This is Dr. Christopher Beyond, Dr. Samuel Galetta, Dr. Elizabeth Emmert, Dr. Philip Anderson, also the undergrads who took hours of their days to really help me out with a lot of this soil sampling and things like that. In particular, a student, Brooke Agnes, is an incredible undergraduate researcher that you should look out for. She helped me with a lot of the packaging of my samples and things like that. Thank you very much, Brooke. Also, I want to thank all the funding that was provided. So most of my funding was through the Salisbury University Green Fund. Also, I had funding from the RAP grant, the Henson School of Science grant. I also want to thank the University of Auckland, New Zealand, specifically the Lear Lab. Dr. Elizabeth Emmert spent her sabbatical last semester learning a lot of these bioinformatic analyses for microbial studies at that lab. So thank you for that. I want to thank Salisbury University in general and the whole Henson biology department. I've had a lot of teachers along the way and mentors that I couldn't have done this without. So thank you so much. And also I want to thank my pilot, Ryan Pappas. This is his single engine Cessna plane. And without this, I wouldn't have been able to get these beautiful images to really complete my story. So with that, I'll take any questions. Thank you very much for being here.
Okay, I don't know if we can clap or not, but
if you turn the sound on.
You can turn off your mics, unmute and clap if you like.
All right.
And then Anthony will take questions.
All right. Hi, Anthony. So, yeah, this is a little bit weird virtually. So great job virtually doing this. I know this is such a struggle for all of you guys as grad students. And let me commend you for a really, really nice job on communicating what you've done. In terms of your project, you know, it was really clear in your presentation, your talk today. So really nice job. Okay, got a question for you. So in thinking about your project and your inferences and how you're sort of thinking about, you know, this globally, I guess my question is, how do you know, so you sampled on Salisbury University's campus in 1 year. How do you know that the particular year that you sampled wasn't an anomaly? You know, how, you know, is it possible that the weather conditions actually really changed the soil microbial content in that particular year, it was not representative of what you normally see. Likewise, why didn't you guys maybe take a sample from someplace like Nassawango Forest as this really undisturbed place as kind of as an additional reference for microbial communities in really undisturbed places?
Yeah, great question, Ryan. And yeah, that's why we did all of our sampling at one time period, and we made sure, so we didn't we made sure that it hadn't rained for 10 days. And yeah, if we wanted to do the most effective way to compensate for these ebbs and different weather and things like that is to do a longitudinal study, right? However, we just decided to do it in one sampling season. And to answer your question about Nassawango, we actually were thinking about that at first, and that was initially what the project was going to be, comparing it to those areas. However, talking about it being in a different region, especially Nassawango, has it's been inundated by water and things like that for long periods at different times. So that's really going to affect these microbial populations. So just for the breadth of our study, we were really focusing on just to see if these communities are different within the campus. So does that answer your question?
Yeah, sure. I mean, I still think it limits your ability to make these sort of broader inferences. But in terms of what you're looking at on campus, yeah, I mean, it's, you did a really nice job with that. So sure.
Yeah. So yeah, especially we can definitely extend upon this study, and there are studies that are doing this. So in University of Maryland, they did this sort of thing in different cities across the globe. So they were in Budapest, they were in South Africa, they were even in Baltimore, Maryland, and they're showing this. So look into some of those kinds of studies. But yeah, this was just a smaller scale study, and we were really just focusing on the Arboretum.
Okay, thanks.
Yeah, no problem. I see you have a question, Dr. Michael Carter.
I think Kim actually had her hand up first. I'm happy.
Oh, sorry. Yeah, what's going on?
Hi. Michael, are you going or me?
I can't hear you.
Are you okay?
You sound like you're in the abyss.
Again, congratulations, right? I guess what I want to ask questions about is the genetics, because you have a lot of genetics students in the audience. And basically, when you're talking about the barcode, I'd really like to know what you're actually sequencing, because ITS should just be DNA, right?
No, well,
I'm just trying to make sure, because if it's an intertranscribed spacer, it's going to be cut out, right? So
no, you're
gene. So that variation is found in those intertranscribed spacers. So if you're using that one and the 16S, are you? So I guess maybe I have two questions. First of all, what did you amplify? Okay, maybe that's the first one.
Yeah, so good question. So we amplified, these different regions.
PCR?
Yeah, we amplify these different regions of DNA. So yeah, just like it's gonna, so on the DNA, it's gonna be expressing these different ribosomes, right? And a part of those ribosomal subunits, we're isolating through PCR. We're amplifying just certain genetic markers within those, either the subunits. So for 16S, we're actually using different variable regions, one variable region in that subunit. And for ITS, that's going to be that internal transcribed spacer, but it's in fact an amplicon product. You have to amplify that from genomic DNA.
Okay, so let me go back one more time. Oh, sorry, I'm getting an echo. I just want to know, so how many PCRs did you do? So per sample, I'm just trying to get the basics. So did you do one for ITS? Did you do one for another? Did you just, so what did you send off, right? So I'm just trying to get the basics, because these are the questions my students are asking right now.
Yeah, that's fine. Yeah, so we pulled those 56 samples, and then we homogenized them and we extracted DNA. And from each sample, we barcoded. So we were isolating that specific biomarker. So we did a total of for prokaryotic organisms, we did a total of 56 PCR amplifications. For eukaryotic, so the fungi, we did 56. And for each of those, we did one amplification for the mock communities that we made as well. So we sent in a total of 114 samples that we were doing for sequencing and getting those sequences back and deciphering.
Okay, just sorry, I'm fighting with my computer with the
It's
mic.
fine.
I just have one more question and I'll let everybody else. Michael, you're next, right? So I guess what I'm trying to get is, I know what next-gen sequencing is, and if you did all these samples, then do you just basically make one giant file of the, so if you're sequencing the next-gen stuff and you have some that's 16S and some that's ITS, are you putting it together to be a barcode? Or I guess that's, I'm going to ask one more, and I promise this is the last one, dude, I know, right? But I'm a market person, probably like Dr. Erickson, so this is kind of important in understanding what the data we're looking at, because if you're doing millions of reads, right, if you're getting all that data, I would expect to see more data plots in your files later on. So I'm just trying to kind of connect the, connect the dots. So
I simplified.
I guess maybe it's because it was sequenced outside. How did they put all that stuff together?
Yeah, so what they do is they do a secondary amplification with a specific barcode for each of those samples. So when they throw it on the sequencing machine, each of those are going to be individually separated based on a certain barcode. So we don't do that. They actually do that. And to answer your question why it doesn't seem like it's too many data points is because that's just for the simplicity of presentation, right? We're collapsing this data, and so it's just representative of just that one sampling point that came from that sample. So obviously if we wanted to show it in a sense that we wanted to show for each taxa or whatever, the screen would just be muddled. You wouldn't be able to make any sense of it.
Okay, thanks. I'll let Michael take it now.
All right, Dr. Carter.
Hey, Anthony, thanks a lot. I really appreciate all of that. I wanted to ask you kind of along the same lines as the same lines of thinking as what Ryan was asking, but taking it in a little bit different direction. How consistent is your data with published data sets, or at least analyses of published data sets? So I think in general, you observe greater diversity and potentially greater abundance in managed locales, managed lawns and gardens, right? The areas that are highly manicured and get a lot of human attention. Is that true across the board?
So good question. So across the board, so a lot of what's going on with this human intervention is they're actually, so when you see the pH is higher and things like that, we're managing it to a point where it's bringing it to a more neutral pH. And these certain places where we're placing more nutrients and things like that and watering these certain landscapes across campus, they're going to like that, right? So if you get food and water, you're going to like that. So in different scenarios, yes, in the studies, they're actually showing these in different areas. However, what's not so clear and why these studies are so important is going down to the microbial assemblage. So just because it's more, it may be more diverse or things like that, it's not actually showing what's happening at the genetic level. So we really need to take this to the next level and see if it's affecting expression as well. But yes, a lot of the taxa we are seeing across, we can compare them to different studies like this. But overall, this field is understudied. So there's not too many studies out there that study specific landscapes within a small area like the university's Arboretum.
Okay, thanks, Anthony.
Yeah, no problem. I see Dr. Patty Erickson has a hand raised.
Sure. Hi. Thanks. Nice talk and beautifully presented in terms of the pictures in the presentation. It really was very easy to follow. And my question has to do with the PCR gel that you showed. And it may have just been an artifact, and it may have just been because it was just a short view, but it almost looked like there was a doublet that you were amplifying in some of those lanes. And I'm wondering whether that's really what the gel looked like when you looked at it, or was it clearly one nice, solid, beefy band?
So yeah, so that was actually one of the gels from ITS, and ITS. So the variable region is around 250 base pairs, but ITS is wonky sometimes. It could be anywhere from 250 base pairs to 600 base pairs. So I was really worried about that too. You see, oh my God, I have multiple bands. What's going on? But yeah, that's really common with these sort of genetic assays, especially for ITS. And there could be a little bit of variability in 16S as well.
Okay, yeah, that makes sense, especially with the ITS.
Yeah, I was, I was freaking out when I first saw that too, and then I called the genomics core, and they said no, that's normal.
Okay, cool.
Any more questions?
I have one more.
All right.
Okay, so I guess it's following up with Patty's now, right? Because the problem with ITS is this idea of concerted evolution, which means that those repeats, because they're variable within different organisms, there could be multiple repeats. And that's exactly what you were seeing on the bands. There could be, the idea is that over evolutionary time, those repeats should homogenize through time and you would get one band, right? That's the idea. But the reason this is studied so much, and it's been a lot of work done in plants, right? And so the idea, if you have a bunch of those bands, right, that could increase your level of diversity for that particular thing. And so have you looked at analyzing, because those multiple band repeats, and it could be 1, 2, 3 in plants, polyploids, they have a bunch of them, right? And so did you have to analyze those species any differently, right?
Okay, yeah, so you're, just to reiterate your question, so the distribution of the lengths of different amplifications, will that affect our analysis of diversity? And the answer is we didn't really have to specifically account for that as much, because it doesn't matter if it's 250 base pairs or 600, they're going to be different, right? And the thing about your question about evolutionary pressures, this is an internal transcribed spacer, so it's between these subunits. So these regions are really good for identifying differences between species, because the differences in them aren't going to really impose, a detrimental effect on at the organismal level, because it's not an actual, so that it's not an actual chunk of what's really necessary for that protein that's being encoded off that gene. So yeah, that's definitely something to consider. And they've actually had some issues with that, because there could, with this variable of ITS, actually like doing qPCR and things like that, there's been some complications, but no, we didn't really focus on worrying about the distribution of those amplicons as much.
I think we're almost out of
Thanks.
time now,
Okay,
so
yeah.
maybe time for one more question at the most. Anything, Anthony?
No, just thank you all for being here, and I hope everyone's happy and in good spirits during these trying times. And I'll be back to Salisbury, don't you worry. So thank you, everyone, for being a part of this journey.
Good job.
Thanks, Anthony. Great job.
Good job. Thank you, Anthony.
Very nice, Anthony.
So I'm just going to stop, and all right, see you guys.
Bye-bye.
Bye.
Nice.